continuing. That's phenomenal, actually.
Yeah, we're really, usually so excited and looking forward for Tuesdays, and hopefully we'll be back with another season.
It's like TV series with seasons. So
yeah.
you need to recharge and then start again.
Okay, looks like we are streaming live. So it's almost 11 or the hour. So we'll let folks trickle in for a minute or two, and then we'll go ahead and do the intro. It's funny, we used to start like 15 minutes before the hour, make sure everything was, set, and we'd sit around and chat for a while, but now it's just like go.
We have gotten better
not as
at
much
it.
time. Well, or just the days are a little more frantic, maybe. I don't know about you, but
yeah, it's always great.
Welcome back, by the way.
Oh, thank you. I lived. My stomach flu is okay.
I'm sorry you got sick with your travels.
It's okay. It always happens when I go after so long. But it was really wonderful. I haven't been home for like a long time, so it's good. I'll go again soon, I think, because this was too short. I barely met anybody.
Yeah, it was a trip for just the important purposes, not forgetting to visit, right?
No, I need a longer vacation.
You're telling me, sis.
Manu's like, yeah, probably didn't have one for like years, Manu.
Yeah, I think I'm excited about the summer for many reasons, including that.
Yeah.
Okay, so yeah, in the name of giving enough time, I think let's go ahead and sort of officially get started. So I just wanted to, I guess, formally welcome everyone to our final cell migration seminar presentation of the semester. It's been 2 full years since we did this for the first time. So I don't remember the exact date, but it was, sometime in the middle of May. We had Jeff Redberg give our kickoff talk 2 years ago, back when the pandemic was new and we still didn't know what the trajectory of that was going to look like. And so we've been doing this for 2 years now, and it's been incredibly rewarding, really wonderful. Yes, and as Adam says in the chat, this is our season 4 finale cliffhanger. I like that a lot. So we're hoping to come back in the fall and keep on doing this as long as people are still interested in coming to online Zoom seminars. I think it's really great because we've been able to connect all across the world in a way that, maybe we couldn't have with just conferences. So we hope you'll come back in the fall. This summer, we'll be working on putting together an exciting lineup for even more talks. So if you're out there and you're interested in giving a talk, just reach out to us, send us an email, send us an abstract, and probably, I don't know, August, maybe beginning of September is when we'll actually be doing it. So send us stuff by July. With that, I just want to thank everybody for being a part of this wonderful, supportive, excited community. And I'll hand it off to Ankita to do the introduction for our illustrious final speaker of the season.
I almost feel like, we're also emotional that this is the last one for the season. Having said that, we are very excited to welcome Professor Manu Prakash. He is an associate professor in Department of Bioengineering at Stanford University, working in the field of physical biology and frugal science, which I'm a big fan of. He runs a curiosity-driven lab at Stanford, combining his passion for basic science while also developing ultra-affordable and accessible technologies that are used all around the world for science education, research, public health, with the goal of democratizing access to the scientific tools. Manu grew up in India, got his PhD from MIT, and was a junior fellow at Harvard Society of Fellows, and was a MacArthur Fellow in 2016. And I would give this floor to Manu, and really excited to hear from you. Thank you
Thank you
so
so
much.
much. It's an absolute delight. I love the analogy to seasons, except the performances. You know, some closures of seasons are not as thrilling, but I'll do my best. I see some friends in the Zoom names. I also see lots of new people. You know, I think one of the big things about sharing science in a very broad way is, you know, these Zoom sessions have been an incredible part. I'm excited that people could connect from multiple time zones. And I'm going to talk to you today much more broadly about, you know, both the role of mechanics in, let me see if I can see my screen. How do I view? It was visible before, but I don't see that anymore. So I don't see anyone. Show video panels. Okay, that's perfect. Sorry about that. I think very broadly, I'll talk about the role of mechanics in information propagation. Go
Can
ahead.
I just interrupt, Manu? Unfortunately, now we all can see the panel as well.
I see.
So
what do I do?
Because it's blocking part of your screen.
I see. What if optimize share? Okay, you know
the only way I've ever gotten it to work is if I had two screens up, and so I don't know if you have two screens or not. But if you don't, it might not be able to work. Okay.
What about
sorry
now?
about that. Yeah, that's fine. There's just the little in the corner, and so hopefully that won't block anything.
Okay. Yeah. No, I think this is reality of Zoom sessions. Okay, so let's get started. You all saw the title. I think I'll go through some of the things that I'm excited about sharing in the role of mechanics in biological contexts. But I think I want to open with this phrase that I've been thinking about for the last couple of years of what I call recreational biology. And what I mean by that is most of you have known about the field of recreational mathematics. And you might have read incredible work from Martin Gardner and many other people that have truly turned this notion of the idea of asking deep mathematical questions in simple, puzzling statements that literally anyone can understand, although it might require, you know, completely reinventing a field of mathematics to be able to answer them. It's grown as a formal branch in mathematics, and I've been thinking about analogies of that to biology. And one of the phrases, you know, as if I was to just very simply give you an example of what I think, from my perspective, what recreational biology might mean, is a very simple question that I want to ask all of you. So if you get bored in this seminar, you could try resolving this question, which is, can toroidal cells exist? This is something that has bothered me for many years, and I'll give you a hint of that in the seminar itself. But, you know, just as a graphics, this is a picture borrowed from one of Martin Gardner's puzzles. If you get really bored from this seminar, you could go try figuring out how many pieces can you cut a toroid with 3 flat planes. But I think more generally, if you were to think about the history of biology and ask this question has been asked before. And if some of you know the context of experiments that were done in forcing dividing cells into toroids, that actually led to a major discovery of how the spindle communicates with the cytokinetic ring, and that leads to coordination in cell division. So sometimes very simple, almost seemingly frivolous questions can give rise to deep insights. And I think I want to encourage that in how we approach biology in as playful a manner as we can, because sometimes in that play lies questions that you might not approach otherwise. So this is a little bit of a philosophical thread that I wanted to touch upon, and I'll bring up the history of the questions that I'll discuss in the topics that I'll care about. The second philosophical note before I start is the incredible people that I get a chance to work with. I'll mention individuals and students and postdocs that have been involved in the work along the way, but I just also want to say plays an incredible part of what we do. We don't take ourselves too seriously. And I think that, I believe, is an important aspect of sometimes uncovering questions that you might be shy from asking. But also, it's just an absolute joy to work with brilliant people. And, I really consider this as a privilege to be able to do what I get to do. So let's dive into a historical context. I'm going to frame this talk in the context of paradoxes. And I think nobody has said this better than Max Delbruck. I just want to read this because I want to share where I'm coming from, almost as an outsider to the context of physical biology. I want to read what Delbruck wrote in some of his writings, which is paradoxical situations analogous to that to which classical physics ran in its attempt to analyze atomic phenomena. This, of course, has been my ulterior motive in biology from the very beginning. And I think what Max is trying to describe here is to sometimes look at paradoxes as fertile grounds for deep ideas and questions. But I think he wrote something in this perspective that even hits to the heart of a lot of work that we do in the lab, which is this tension between biochemistry and behavior and physical context. What should you attribute to biology? What should you attribute to physics? And how much of that is actually broken in its framing itself? And he wrote, the physicist, may be told that the only real access to atomic physics to biology is through biochemistry. Listening to the story of modern biochemistry, he might become persuaded that the cell is a sack full of enzymes acting on substrates, converting them through various intermediate stages and waste products. And many of you know that that story is only part of the ensemble, and this is not how we think about modern biology. But it's important to kind of look back when molecular biology was at its height, when many of these sets of things were literally being discovered. Everything was attributed to a bag of enzymes. And it looks sane until the paradoxes crop up and come up into sharper focus. In biology, we are not yet at the point where we are presented with clear paradoxes. And this will not happen until the analysis of behavior of living cells has been carried out into far greater detail. And I think this is what I want to highlight. He really emphasizes the idea that study cells in their own terms. Try to understand what they do and derive theories from those sets of behaviors. I believe that it is in this direction that physicists will show the greatest zeal and will create an intellectual approach to biology, which will finally lend meaning to the ill-used term biophysics. There is a mouthful in here. There is a lot that I could say that could be controversial to people, but I just really love the approach of being able to study systems as a whole. And from that perspective, I'm going to talk about behaviors of single cells today, and talk about the context in which the environmental context in which these behaviors occur, and go back and ask what role does mechanics play, especially in the context of mechanical communication, mechanical computation, and mechanical control of these sets of phenotypes. So many of you know the history, at least my own history in science begins from mechanical computing. I got a PhD at MIT studying bubbles and building little computers out of bubbles. And I've pursued that passionately in trying to build and think about new materials that could be used as means for mechanical computation. We have done this in the context of, literally drops and bubbles. We have recently done this in the context of an adaptive material we call it active foam. Much of that is published, but I want to allude towards this paper that we finished in 2015 that has guided some of my thinking in when I'm thinking about cellular behavior, for example, in which actuation and sensing could literally be incorporated in a single mechanism that could lead to fairly complex behaviors that you might always attribute to biological systems. This is a growing field of active matter and active droplets. But I love this analogy in which literally food coloring on a glass slide, this would be my example of a simple frugal science experiment that you could do at home to explain somebody how chemotaxis or cellular motility might actually work. And this is all published work, but the only thing I want to emphasize here is when you deeply think about actuators in a non-equilibrium context, they themselves, because of their sensitivity to the environment, can actually become sensors. And when that happens, truly remarkable phenomena occur at all length scales. One example of this is, of course, a sense of self. There is lots of really funny things we were able to do with this system. And more recently, we've actually also shown several years ago that that same system is capable of self-reflection in the context of being able to see your own vapor gradients. And so you can see up on the panel on the top, two droplets attracting each other. You can explain that from the context of thinking about how a droplet modifies the vapor cloud, or from, in this context, thinking about the information cloud that might exist around a droplet. But if you were to just have a barrier with no droplet at all, that is sufficient to break symmetry in the system, as if the droplet was actually seeing itself in the mirror. And what I was delighted to see a couple years later in the neutrophil world, much of these ideas came out to be true. And now we do understand how cells can actually look around the corner based on ideas of how these sets of diffusion clouds might actually be perturbed. These chemical gradients could be perturbed by their environment alone. So that is sort of a short attribute to thinking about abiotic systems and how abiotic systems might inspire us. But today's talk is primarily going to be about biological systems. And I want to jump and really focus much more on single cell behavior. But one thing that I do want to mention before any of the single cell behavior that I'll mostly talk about is our work in multicellular context, which is something that's now published in this trilogy that I call of the question of a mechanical brain, which is thinking about how animals that do not have neurons could potentially exploit mechanical information processing as means of fairly complex behavior. And because much of this work is published, I want to talk about completely new insights in terms of work we're doing now. But if some of you are interested, there is a little bit written about this and more to come in that space. So let's start with the menu for today. I want to talk about that puzzle that I mentioned to you, whether toroidal cells can exist. But of course, since this is a cell migration seminar, I'll talk about this in the context of what I've been thinking about as the longest cell migration on the planet. And I'm actually curious if people on the chat want to write about what might be they think is the longest cell migration. I'll jump into talking a little bit about dissipation in cells in the context of topology. And then if time permitting, I'll try covering these two ideas of topological traps and geometry encoding behavior. So let's start with the longest cell migration. I'm curious if people have an intuition, how might the longest migration in a cell be? Could it be the size of an organism, which you could believe a couple of meters, or might it actually be longer? And since I don't see chat answers, I
can read them to you.
This might be fun. Let me just open it for a second. Neural crust in a whale. I absolutely love that. But I think Buyang got it right. It's the plankton in the ocean, which is really what I'll talk about. But I have to say, Sharon, the creativity of, I did not think of the neural crust in a whale. And just, much of this is unfolding in real world, except we have absolutely no idea what those sets of limits look like. But I'm going to tell you about a cell that migrates roughly around 200 meters fairly regularly between a day to a couple of days. And this really comes from the context of the ocean. This has been a big thrust in the lab with a big team, which we now call Gravity Machine Team, focused on thinking about role of gravity in life in the ocean. And this particular work is spearheaded by a postdoc in the lab, Adam, and another postdoc in the lab, Rahul, with a lot of support for Hong Chan and several other students that have been working on the tools that make this work possible. And the visualization that I want you to have is, imagine walking into the canyons in the Grand Canyon, and you can walk into a chasm that's been cut into history. and you could literally see through life that might have existed in the past. Similarly, imagine if there was a chasm that was cut into the ocean, and you could literally see life layered through across all different heights. And this is something that we very passionately think about because of the context of diel migration. It is a fundamental process on this planet, and we've been working on this process for the last 5 years, asking the question of how cells make decisions to migrate at such long distances. Now, this is only an animation, but there exist real data on sonograms and using sound to detect density of biomass. This is a diel vertical migration where literally plankton could travel kilometers, and even single cells can travel all the way to 200s of meters. And what I find fascinating about this process is many of you have admired Serengeti, but literally this is in biomass much larger than that, but happening every single day. Every night you go to sleep, the process where you can see the shade of the sun traveling and vertical movements in the ocean. And if you do the energy budget, roughly, the commute energy cost of this is actually the annual energy budget of United States, which I just find baffling, giving an example of cell migration, of what incredible biology and its context to geobiology that cells can do. Now, why is this important to think about? I've been championing for a while now that as cell biologists, we need to deeply think about climate change. And the reason this is important is because this is incredibly linked with the process of how the ocean actually sequesters carbon and has the capacity to lock carbon for a long periods of time. This is called the biological carbon pump. And just to give you an attribute, just to think about, how do you study migration and cellular motility that's happening at this bizarre ecosystem, which we have so little tools for, and at the density at which it's occurring? This is a real footage of a deep sea black diver doing a dark night dive and being able to see the kind of intensity and density that the life is interacting itself. It's very difficult to actually study in its native environments. Now, the other reason associated with understanding this pump is if somebody was to switch this pump off, carbon in our atmosphere would jump from 400 to 600 parts per million, which is quite a scary scenario in terms of thinking about tipping points that many people have talked about. So the question that we're going to ask today is, just take any cell, and for fun, I'm going to choose a non-motile cell, or a cell that we had assumed until the last 200 years to be non-motile, and show life cycle in the context of ecology. And we will do this in a perspective of this large-scale migration, but also thinking quite a lot about the physical phenomena that have to underplay. If you could be in the frame of reference of this cell, what would you actually see? And to just give you a little bit of a better context for this, for some of you who don't think about oceanography, imagine your landscape of gradients, but imagine, literally every possible parameter that you can think about is vertically stratified in the ocean. So the oceans can be much more homogeneous in the XY plane, but in the Z plane, they're incredibly heterogeneous, all the way from starting with very simple thing like light. For a couple 150 meters, you actually enter darker waters, which is the mesopelagic zone. So if you are a phytoplankton, a cell that depends on light, you better be up there. And I'll talk about this in the context of what I call gravity traps. The other context here is the sinking and the sedimentation. The density of water has a gradient, the salinity has a gradient, the temperature has a gradient, nutrients have a gradient, oxygen has a gradient, and on and on and on. And cells are making decisions associated with this for where they want to be. And of course, some of these gradients are temporal, for example, light, or when upwellings arise, when nutrients change over time. And the question that we want to understand fundamentally is, how on earth is it possible? Now I'm referring to the paradox that I talked about, a non-motile cell for hundreds of millions of years to survive in this context, when literally the cytoplasm of most cells is between 5 to 10% the density of seawater. How on earth is it possible that this damn thing, this doesn't sink to the bottom of the ocean? over hundreds of years or thousands of years for that matter, and especially for something like a diatom or a dinoflagellate that's fundamentally dependent on light. And seemingly the paradox here is that the cells do it not just with ease, but they do it in the context of being labeled as non-motile. And we started studying this, and this has baffled us for the last 5 years until we started literally just watching cells. As any good cell biologist will tell you, whenever you have a paradox, just take a back seat and watch what cells do. So that's what we're going to do for the next 5-10 minutes. And the cell that I'm going to choose is actually a cell that you have all enjoyed. I know many of you have roots into MBL, and many of you have enjoyed bioluminescence. So we will talk about a deep diving non-motile dinoflagellate called Pyrocystis noctiluca. This is responsible for an incredible bioluminescence that you've all possibly had the chance to swim in. And if not, I would highly encourage, if you want to see a magical phenomena for cell biology, go play with in bioluminescent waters. Now, what's fascinating is in Swift all the way in 1975-76, noticed a paradox, which is he was able to pull out viable cells, not spores. These cells also form spores, which I'm not going to talk about. Viable cells from 200 meters deep, that is right at the edge of the euphotic zone. And you might wonder, if that cell doesn't have the capacity to rise all the way up, how on earth in generations, you can think about the amount that you sink, you have to have the capacity to be able to swim up. Otherwise, you would really, leave the ecosystem forever. And of course, there are certain kinds of upwellings associated with the ocean, but the horizontal velocities in the ocean are literally multiple orders of magnitude smaller. One way of thinking about, for some of you who think about turbulence, that in these sets of depths beyond the mixed layer of 10, 15, 20 meters, the ocean can be calmer than the amount of turbulence that you generate when you stir your cup of coffee. So you cannot just rely on passive oceans to be able to circulating this. And this is why it is very well known in oceanography that the ocean is not a well-mixed bath. The question here is fascinating because we need to be able to study cell biology, but we need to be able to understand that in the context of literally at least hundreds of meters. And as a lab, there is a long story about how we got into this problem. We were studying parasites in Madagascar. And I remember one of my early students at that time, Deepak and I, we were packing to go to Madagascar, and we had built a very long microscope. Our goal was to build a 100-meter microscope, and we had built a 10-meter microscope. We were packing tubes. And while packing tubes all the way to be flying to Madagascar, we had an aha moment, which is, what if the two ends of the tubes are actually connected? And that led to what is called Gravity Machine, which is now a big team in the lab trying to build this technology to understand long-term migrations of aquatic cells in as native an ecosystem as possible. And the simplest way of describing this is literally a gerbil on a wheel. So think about, but this, rather than this being animal, this being a single cell. And, this was not just a cartoon. It took us a couple of years, but we built a technology that now allows us to track and watch cellular scale motility at length scales, actually at infinite length scales in the z-axis. And since I'm not going to talk too much about the technology, I want to talk about cell biology. The simplest way of thinking about it is take your normal microscope, tilt it 90 degrees, and put your cells in a glass wheel. This glass wheel is roughly the size of my hands. And now when a cell is traveling up, we move the wheel down. And when the cell is moving down, we move the wheel up. And so for many of you who care about hydrodynamics, you might be scratching your head to say, no, that's not possible. You're going to induce inertia in this system, and that might perturb what the cells do. And we very carefully mapped, and it turns out that nature was kind to us, that in the viscous regime, we were able to map the rotational flows in these systems to really come up with a criteria for what we call behavior velocities of migration associated with single cells. And as long as this condition that I write in this green box is true, we can truthfully track a trajectory of a single cell traveling at infinite distances. while in the lab frame of reference, the cell is actually stationary. So I could really do cell biology and study this in a subcellular context. And not only do we take these machines and build them in the lab, we actually take them to the field. And this is the discovery that I'll first tell you about of the long-term migration of Pyrocystis. What you are watching here is essentially a boat trip that we took on Kilo Moana. This is hundreds of miles off the coast of Hawaii. You know, science can be adventurous, and this was truly adventurous. And to explain to you what I mean by adventurous, this is us collecting data from a cell that has been pulled out from 75 meters deep on a CTD rosette that I'll show you, brought into a lab setting, immediately transferred into this virtual reality environment. And I'll explain the virtual reality in a context, because we can artificially induce the kinds of environment that now patterned on the z-axis. But the reason I want to point out the adventure is while we were literally collecting this data, we were 2 hours or 3 hours away from a massive storm, and we had to buckle down, and actually the ship had to turn around because the weather became dangerous. But in the mile, the weather was not dangerous. We were able to collect some remarkable data. That's really the rosette where we collect the cells from. This is the deck of the ship. And it's really transformative, at least I have to say, being out in an environment that this cell biology is occurring to truly understand this context. And this is the kind of data that we collected, where at different times we would find cells that are sinking, and at other times we would find cells that are rising. And some of you might already notice that there are certain morphological changes that are already being visible in this context right now. And I'll explain that in a little more detail. But this was the starting point of the paradox, is here is a cell that seemingly has no flagella or no swimming appendage per se, and it has the capacity to migrate such long distances in the ocean. Now, of course, we had to bring this specific cell in the lab. We started culturing it, and we noticed another surprise, which is this massive explosion, or what we call ballooning, that happens in this cell during cell division. So what you're watching here is this Pyrocystis noctiluca cell. Right after cell division, you notice that the cell starts inflating and keeps inflating and keeps inflating until around 6-fold of volume change. And you should be scratching your head, because remember, I mentioned to you about the enzymes associated. If I was to dilute any enzymatic reaction 6-fold, how on earth is it actually possible for any biochemistry to still be viable? And that remained as a paradox for a while, until we started thinking about topology and toroidal geometry. So first of all, here is on the left is some data to show that cell expansion essentially changes buoyancy. And for some of you who saw the Stokes law that I had previously described in the context of sedimentation, because it is a function of radius, you can see that ballooning can change your sedimentation velocities. But the puzzle gets even more interesting. And one of the important aspects of this for us was to first confirm what is the life cycle of the cell, and can we map this life cycle to the ecology of the cell? So this is why we term this work ecophysiology, which is now, this is 7 days of a time lapse of a culture of cells. And you notice that this is a remarkably repeatable phenomenon. Every time, this is day and night, you can see the plastids, the chloroplasts being deployed in and out depending on the time. When the day comes, the plastids go out like antennas, and in the night they come back. But at that massive moment of event, there is this massive expansion that happens in all cells. And again, we were able to recapitulate this in the lab, because I was a little bit worried that the phenomena that we saw at sea might not recapitulate in a lab strain. And so here is a big question, is, as a paradox, how do we really think about this cell dilution? And if this is actually a mechanism for motility or migration, how would you even think about this for the cell to still be viable? And the paradox got resolved when we started doing light sheet imaging on these cells. And this is the architecture of the cell. Now, tell me this is, not a beautiful cell. What you're looking at is essentially a N genus torus. which is, so the way to think about it is, take a donut and make thousands of holes in it. So the nucleus is right in the center, but the cell has adopted a really strange topology. And the topology is that all the cytoplasm is in these tiny strands and threads that form a net-like structure that is around a vacuole. And much of the expansion that you actually saw is in the vacuole itself. And of course, this is a dynamic network that we're very excited about trying to understand how these dynamic networks remodel. But from a topological perspective, this is a TEM of this, when you can see the plastids transporting through these tiny networks of membranes topologically connected to the nucleus. And this is light sheet data associated with that cell. And you can see the massive expansion while preserving the actual volume of the cytoplasm. What Adam was able to do in the lab is to actually quantify every one of these volumes to demonstrate that the volume of the cytoplasm does not change in this dramatic event. And at that same time, we were able to build an entire life cycle of the system associated with this cell. Now, it's fascinating to think about how do we map that life cycle into the context of the ocean. And one of the important aspects that I'll talk about now is really deeply thinking about cell cycle and cell physiology in the context of an ecosystem that it might be in, whether that ecosystem is the open ocean or aquatic, or whether that ecosystem is actually the tissue or the body that a cell lives in. Because it's incredibly important to be thinking about these cues from the context of evolution of cell cycle itself. So I'm going to divide this cell cycle into this division phase that leads to a very rapid expansion. That expansion is roughly 8 to 10 minutes. Then the cycle turns into growth, which is increasing protein production and actually increasing the cytoplasmic density. And that then transitions into what we call pre-division, which is a third vacuole gets separated before the cytokinesis ring occurs. I'm also not talking about how this cell divides. That's another pursuit in the lab. But some of you who think about cytokinesis should be puzzled by the fact that how do you make thousands of cytokinetic rings? And we have an idea associated how physical context might actually enable the cell to do this. So let's jump a little bit to some very simple mathematical ways of thinking about these approach. I want to write down a minimal 1D vertical migration model for something like this. So you can see here just the sinking velocity written. And now, of course, you can see I've written this term phi, and you should think phi as an activity parameter as you transition into the cell cycle. So you can arbitrarily define phi from 0 to 1, and wherever you are defines the cell cycle. Now, what's beautiful about this system is that the cell cycle has to be closed. Hence, it's actually important to be thinking about these parameters to have a closed boundaries. I've written down the effective density, which is associated with the effective density of a cell as a function of cell cycle. And I've also written down the seawater density because, again, it is in a patterned environment of density, which is the seawater density is patterned as a function of Z. Now, the beautiful aspects of this is we know what the pattern density, which is called the pycnocline in the ocean looks like. And you can see a very sharp transition around starting 100 meters or something when it starts to crash dramatically. While we also know that there is internal behavior, which is both the radius and the density are programmed by the cell. And we can put this in to write down a dynamical model for Z height as a function of cell cycle. And I don't have time to go into this, but one of the things that we can do is we can non-dimensionalize these terms in the context of minimum radius. And that leads to a final equation, writing essentially the closed boundaries for this to this term, which I absolutely love in its simplicity because it allows us to understand this question of what we are now calling the Z phi space, which is how can I define a cell in its environment and where it will be as a function of its cell cycle. So cell cycle itself is encoding where you would be in your patterned environment. Now, very important to point out here that I have not included the feedback loops that must exist in the system, because remember, if you go below the euphotic zone or you go below the critical depth, which I'll show in a second, you might end up not making the distance up, and slowly you could actually disappear from the environment, which is what we call a gravitational trap. And because gravity has always been on our planet, we really have to think about this trap very carefully. But without including feedback, which would essentially increase or speed up the cell cycle or slow down the cell cycle in different ways, we can still write down a term which has this beta Z term, which is associated with what's happening to the density of water, and has the phi and the R phi term, which essentially is truthful to the context of thinking about the physiology of the cell. And a single equation then describe a trajectory of a cell as a function of a spatial environment mapped to the cell cycle. So you can think of this as a spiraling staircase. In certain cases, these spiraling staircases have the capacity to remain in this euphotic zone. So for example, I chose 2 different parameters, and I started that cell at the same depth of 100 meters. You can see one of them slowly climbed up and remains in the euphotic zone, while the other one cannot. And what's even more fascinating is you can put these sets of terms to write down a phase space of what we are now calling a gravitational trap, which is there are certain sets of parameters. And if those sets of parameters are true, which start with the initial cytoplasmic density, which I believe must have certain constraints because proteins, you cannot dilute them infinitely, you would end up in a gravitational trap, which is this red curve that no matter what you do, you will always sink eventually unless you are helped by the environment, which is the upwellings that exist. But because these cells are persistent in all waters in the ocean where there is no upwelling that is known, because upwelling is known primarily closer to the coastal lines, you would find this cell to be able to persist. And what I find fascinating is that number that's associated with this peak gravitational trap is actually close to the maximum cytoplasmic density that we find. Of course, there is much more to it in the context of evolution of a cell, but what we do believe is gravity played an important role in trying to understand the fundamental limits that might exist on how dense you can make your cytoplasm. Now, the story goes on, and I don't have time to dive into it further, but this is something that we will post soon, so I would be happy for people to comment on this, but this is unpublished work with Adam and Rahul and a couple other people engaged in the lab. I do want to leave a puzzle on topology before I jump to another paradox, which is what I find fascinating is, of course, topology came to our rescue where cell architecture was fundamental to its capacity to balloon so fast to turn around at a critical zone where it was very close to the gravitational trap. Now, what's even more fascinating is this happens to be a cell that also has a toroidal nucleus, which is absolutely bizarre to think about, that microtubule structures actually spear through the nucleus itself to turn that geometry into these cascading castle structures. And of course, we have been trying hard to actually image this nucleus in real time to be able to understand how do you undergo a cell division while maintaining this topological context. And as you undergo multiple divisions, you have to also worry about maintaining the N genus, because every time you are splitting this toroidal structure, you are reducing the topological, the number of genuses involved. So there is a lot more to this puzzle than what I have talked about. And actually, just as a closure on the oceanography before I jump to land or much more aquatic freshwater systems, this is a general principle. I talked about dinoflagellates because it was a viable system for us to study in deep waters, but we've been also studying this in the context of diatoms. And most of you know 30 to 40% of all oxygen on this planet comes from diatoms. And I find it incredible how little we actually know about the life of diatoms. This is in the context of cells that live in a glass house, and because of that, seemingly do not have any cilia or flagella. It actually turns out in a sexual phase, there is a category of diatoms, especially centric diatoms, that do have a flagellated sperm, but that's a very small portion of its life cycle. But we're going to talk about a puzzling data set that we've been working on for many years that we discovered in Puerto Rico. And I absolutely love this data set because, talk about paradoxes. This is from centric diatom. You're watching this data set in the gravity machine. And I just want you to observe this. here is a cell that is made out of, a silica shell. So it's heavy. And so it sinks, no surprise. But wait a second, just watch that trajectory. There are no interactions with walls. This is deep in the water. And suddenly out of nowhere, as if a stone could start floating, within 100 milliseconds, the cell has the capacity to stop its downward migration and just hang out like a Buddhist, stone, things that I've just seen in movies, except, this is real data. And then suddenly it says, I don't like it here. Let me sink again. And then suddenly a fraction of a second later, it stalls. We call this blink behavior. And what's fundamentally happening is the cell is essentially perturbing its density in hundreds of milliseconds. And this is a phenomena that we have observed across a large number of cells, both caught in the wild, in lab settings, smallest diatoms all the way to 5 microns to largest diatoms that are 500 microns. A very universal phenomena. And we've been thinking quite a lot about, again, doing the same thing that I described in the context of now the cell cycle and motility of a cell associated with its environment, but now connected to the context of a diatom, which is doing this behavior even at a faster scale. I'm not going to talk too much more about this because I want to touch upon one or two other news stories that are associated with cell migration. And actually, it might be just useful to take questions in the very end. So if this is okay, Ankita, I'm going to switch to another topic and then come back to questions. Does that sound good?
Yeah.
Okay. I think I was thinking I'll have time to tell you about hydrodynamic trigger waves. But looks like we're going to run out of time. I want to tell you about another story that's unpublished. And so since you can't read a preprint yet, it might be fun to tell you about here, which is in the context of what we think as active cellular motility combined with the context of what we call topological traps. So this goes back to thinking about spirals. And you have to kind of change your context, but keep the evolution of life on the planet in mind. This story is about cyanobacteria. As many of you know, oxygen is the greatest poison on this planet, at least if you ask the cells 3 billion years ago. Oxygen pumped into our atmosphere was one of the biggest dramatic change that happened, killing literally massive amount of life on this planet. That all happened because of cyanobacteria. And one of the fascinating things that we've been thinking about for a while is what about the context of what cyanobacteria could do led to this massive dominance on this planet. And again, one of the attributes that is given in this perspective, of course, cyanobacteria have been around 3 billion years ago. Chain-forming cyanobacteria started forming all the way to 2 billion years ago. But only 1 billion years ago or 0.5 billion years ago was really this massive transition. It took a while to pump oxygen into the planet. And that must have been at a time when these sets of stromatolites dominated the planet. And now one of the phenomenal aspects of stromatolites is these types of roundish structures that they form. And there have been certain sets of theories and ideas. And we started asking ourselves, what about cell motility and cell migration might enable these structures to be so stable? And if you go back to fossils and certain early fossil structures, you can find these spiraling structures that exist in the fossil records. And of course, these are not just single cells, these are thousands of cells lined up in chains, but then these sets of chains intertangle and intertwine together to make these persistent structures. What we believe is that this persistence might be valuable in terms of establishing a niche and establishing kind of a presence in an ecosystem which was changing fast, such that ecological parameters don't change over time. So we started studying this, and this is work of a graduate student that just finished Ting Ting Gong. When we started looking at this almost 7 or 8 years ago by an accident that I made when I ordered the wrong cell type, I was interested in trying to understand bubbles inside cells. But unfortunately, somebody without me knowing changed the name of the species, and I ended up with oscillatoria in the lab. And it turned out it was okay because we ended up discovering a new phenomena. The phenomena is this. This is cells that we plate on a soft gel, and we just watch them. And because I have short time, I'm just going to stretch this. Over a period of 5-6 hours, these sets of cells form patterns that you can start seeing these circular round-like spiraling structures. And I'm just going to go to the very end to show you how consistent these patterns are. So you can see on the right, and then on the left is just one image of these sets of patterns. And again, mind you, today I will only talk about the two-dimensional context of this problem, although we are working on the three-dimensional analogy to this. I found it remarkable to think about how stable these structures are. And I remember leaving the bottle outside my office, coming back a few days later when I had ignored my cultures, as we all are supposed to do, and finding these incredible patterns all across these bottles. Now, the quest became, can we understand the stability of these structures? Now, these are cells that are very long chains, but they have a remarkable emergent behavior, which I'm not talking about today, which is the association of reversal. So you can see small filaments of cells are gliding across, and then they have the capacity at a certain frequency to reverse. We understand quite a lot about this reversal, but not everything. So I'm not going to talk too much about this, but just want to give you a hint for how do you go from single cells that could be crawling around to these massive spiraling structures. And the notion of spirals will become important because spirals have existed at all length scales. I'm sure some of you are wearing the traditional watch. There is a spiral spring. But if you go at the smallest length scales, you can find DNA spirals. You can find spirals in the escort machinery that exists in pinching the sets of membranes. And of course, for some of the physicist friends in my room, we are literally sitting in a spiral, which is our spiraling galaxies. These sets of spirals, I want you to focus in how we will define that the spiraling galaxy that we are fitting in is entangled group of filaments. And we will talk about this entanglement briefly now. First of all, how does this cell move? The current theory that's out there, which is really a lot of work that has gone in, is kind of a jet propulsion theory, that these sets of cells ooze out a mucus, which is directional, and that gives rise to directionality, while directionality is coupled to chirality, where the cell is actually also twisting as it's moving along. But I want to focus much more on the large scale phenomena. So first of all, these sets of spirals are incredibly stable. We can have the spirals if we don't let our system dry out on a microscope. We can have them for days. But you notice something really interesting. It is not just a traditional vortex of all cells moving in the same direction. It has flux in the tangential directions that are intertwined. You can watch filaments that are moving clockwise. You can watch filaments that are moving interclockwise. And this entire system is actually breathing. And after some amount of work, I love this movie because it's almost hypnotic to be able to look at. Ting Ting was able to demonstrate that the flux in this system is going both clockwise and counterclockwise, while the same system remains incredibly stable. And of course, the filaments still undergo reversals, which is why it's possible. So choose your favorite filament here, and you will see that it's actually moving forward and backward. And at some point of time, it's making certain sets of decisions. And these sets of decisions could be mapped into reversal frequency as a function of average length of the filament, which is associated with the idea that longer filaments take a longer time to reverse. And you can think about this in the context of mechanical information propagation, which I'm not talking about today, but we have mathematical models to be able to explain that type of distribution. But what I'm really fascinated by is not only is there radial flux, not only is there tangential flux, there is actually radial flux as well. If you notice one of the pink filaments in this set of a movie, you will find that the filament is not trapped. It is able to go all the way from the inner edge to the outer edge of this spiraling active structure, which is seen in this chymograph. And that can only be possible if these cells can entangle into each other and make way. And of course, we can verify that this is purely a two-dimensional system, so there are no crossover points in these current data sets. And what's beautiful about this analogy is when we make the analogy to this to dislocation theory. So many of you who have taken solid mechanics classes know and understand dislocation theory, except I will talk about dislocation theory in the context of a polar coordinate system. And what I want you to focus on is a very simple Burgers vector associated with this, is think of a polar lattice, and every time there is a filament, it leads into a Burgers vector where there is a jump between what has to happen. And every one of those could be thought about as a dislocation defect. Now, I can also map this in the context of the energy, the elastic energy that's stored in these sets of defects. And now we could possibly watch this dynamics in the context of defects alone. There are lots of examples of thinking about dislocation theories in layered solids. So we borrowed some of the example. And the simplest way to think about is shuffling the deck of cards. I know all of you love playing cards. So take a deck of cards and shuffle them together and watch them from the side. What you would notice is what I'm doing with my hands, that you have created a massive number of defects. But now this is not any kind of dislocation. It's active dislocation theory because these filaments are actually active. And what that leads to, in which I'm mapping here every one of these dislocations, we started asking ourselves, could we actually understand the flux dynamics in this system purely in the context of how these defects interact with each other? Now, first, to map that, you can just visualize these sets of defects, and you can see they annihilate each other because there is a moment of time that a positive and a negative Burgers vector could join together. But in a traditional context, that defect would have disappeared if this was not active mechanics. But because this is active mechanics, the cell motility is strong enough to overcome the energy barrier of creating a defect. These sets of filaments that come together can remain together for a little while, but then slip past each other to create the defect again. And that leads to a conservation of defects in this context, that the total number of defects is twice the number of filaments, and that will remain preserved. Now, one
Manu,
of the other things that we can do.
Sorry, Manu, I'm interrupting because we're getting very close to the
Okay,
end time.
So let me just show the very last movie, and I'm going to end in less than a minute.
Sounds good.
Unfortunately, I have run out of time, but let me just jump to the paradox resolution. First of all, we can explain this set of a dynamics in the context of a class of rules associated with this defect dynamics. We can write down this as a context of rules, and then we can actually map this flux in a context of a computational framework. Now, if that was not puzzling enough, I just want to end with this puzzle, which I said, why are these structures so stable? And that bothered us for a long while, because although we were capable of explaining this set of a dynamics, why doesn't this thing fall apart? And then we discovered a remarkable mathematical properties of spirals. So I want to close full circle with recreational biology, which is you can doodle this yourself to find out, which is what we call topological traps. What we noticed, there are certain types of structures that lock their cells, that there is no radial flux that happens, which you can see in the bottom. While there are other structures that have flux going all the way back and forth. And the fundamental phenomena boils down to what we call geometrical chirality, which you can explain yourself in a couple of minutes. If you take a clockwise spiral and a counterclockwise spiral, so this is one of the counterclockwise spiral, this is one of the clockwise spiral, and then you interweave them together, no matter the amount of activity, it is not possible for them to cross over each other. So the moment you add a different geometrical chirality as a new filament that's coming in, suddenly the structure inside it is locked. And we're calling this idea topological traps associated with cells, because this is what leads to a phenomenal stability in these structures, because no matter the amount of activity, you cannot cross that barrier. And what you see in that picture on the right, which is with these purple and the green lines, is those are the fundamental topological traps that no material can pass. And so cells can do all kinds of things inside that trap, but cannot cross over that barrier. So I'm going to stop here. I just want to share, we've been very excited about this work in the context of thinking about active spirals with dislocation theory, and especially active dislocation theory. and a discovery of a really physical, something that could have been discovered in the context of recreational mathematics, but we discovered it in the context of recreational biology. I'm going to end here. I apologize for going way over the time. I just get excited. I have some time, so I can still chat if people have questions.
Sure. Thank you so much. I mean, I guess I have to read a lot about this to absorb. I'm going to take a question from Byung. Says pressure increases, and this is from the first part of your talk. Pressure increases as plankton swims down into water, and the effect of pressure may be significant when the travel distance is in the order of tens of meters. So he doesn't understand how your wheeled device can recapitulate this, but do you think this pressure change can also be important in biophysics?
Yeah, I wish I had multiple hours to talk about this, because we've been working on this wheel as a simple idea of a tabletop ocean. So not only can we recapitulate actually pressure, we can recapitulate light, we can recapitulate salinity, we can recapitulate chemical gradients, for example, nutrients. And the simple way to think about it is we take spatial gradients and we turn them into temporal gradient. So we have a syringe that's attached to the wheel that essentially turns the pressure of the wheel constantly higher. And as everybody knows, 10 meters is 1 atmosphere. But it is in the context of a cell, it's a virtual gradient, because as a cell travels and the wheel turns a virtual distance, it is perceiving a pressure that is higher or lower. And not only that, what gets really exciting is, because this is true metaverse of cells, we can flip what exists on our planet. We can make pressure decrease while the cell is going down, and although gravity is pointing downward, and a cell in its entire life, in the history of life on this planet, has never seen a cue that's backwards. So this is, again, very important in thinking about when you have sensory stimuli that are coming from the environment, they can be confusing sometimes. And so we can build a weight function. This is almost like playing a hand of God a little bit in changing the environment in a manner to unpack lots of these feedback circuits that we're excited about. And again, there's a lot of work that we do on the context of pressure, but just not enough time to talk about it right now.
Thank you. And before I take the next question, I just want to say that I think that this talk has really inspired all of us in the audience to remember why we became scientists in the first place, right? Because the world is fascinating and beautiful and we love it. So thank you for that really fantastic and wide-ranging talk that's actually pointing out to me lots of things that I don't know. So our next question from Ashish Biani saying, enhancing surface area without increasing cell plasm volume would help a cell utilize upwellings, which, as you said, are not available in a lot of the ocean. So wouldn't additional buoyancy be needed to be created to travel up, maybe by sequestering gases in the cell?
This is a very tricky point, and it all got rushed into the mathematics. It took us a long while to figure out. So you can slow down, but to be able to rise up, what you have to do is change the effective cell density. The effective cell density includes the effective density associated with the vacuole and the cytoplasm. And the way the cell does this, we just finished the transcriptome for the cell, is we have discovered several novel aquaporins in this system. And what the cell is doing is using aquaporins. This is what the inflation event is. Very much like, I don't know if some of you know the history of aquaporins in mosquito biology. Very similarly, what the cell is doing is a massive influx of water. And it's able to choose that influx of water to be able to change the net buoyancy to actually be lighter than seawater. And we can shut down several of these ion channels to really demonstrate that the entire process of inflation pauses until we reflush the system. And then suddenly it's almost as if there is a pause and these cells were waiting to inflate. And then suddenly the cell can inflate again. So there is a phenomenal molecular context of this, which is really the biology of aquaporins, which I didn't get the time to talk about, but it will be in the archive paper that we'll post.
That is so fascinating because aquaporins have so much context in cell migration as well.
Yeah, no, I mean,
They're everywhere.
they're everywhere. And there are several novel structures that we have that we are still trying to understand.
Awesome. Next question is from Beatrice asking, she's curious if you have found any connection between the geometry of diatom shells and the ability to change their density.
Yeah, I think this is literally, I just got back from the Gulf of Naples, and two of my students are in the Mediterranean right now, where we are mapping cell geometry to cellular sedimentation using this tool of fresh diatoms that are collected right out of the water. One of the challenges that we have seen in cell culture, and this is again a warning to many people who think very broadly about cell biology. When we study behavior, we find within a couple of generations, the cells that we bring out from the environment let go of the wild behavior. And it should not come as a surprise. We are bathing them in nutrients. They don't need to do migrations. And so we are very careful about this, but there is a phenomenal context of geometry. One simple example is a cell that we study, which are the Kurtosauruses, which have massive whiskers. So almost sometimes 30 to 40 times the size of the cell. The cell is only 5 microns, and these whiskers could be hundreds of microns. And these whiskers, just like a mustache, except a mustache of a cat, essentially reduce the drag associated with sinking. And we are just starting to get to a point of building a universal map of cell geometry and sedimentation. But I'm very careful talking about that data because we really need to make sure that the data, because the cell is a living object, and we can do this for frustules and other things where we can purely understand the role of geometry, but we have also discovered the cell is capable of doing 100 millisecond fluctuations in density. So you have to be very careful about making sure that the conditions, the cell is not stressed, because if the cell is stressed, it might make a choice, and then you would not have the kind of statistics to connect geometry to behavior.
Fascinating. We have a question from Raphael Petrosian saying, thank you for the very insightful talk, and wondering, have you tried sinking experiments with the diatom cells, the ones that showed the two-phase sinking behavior in the following environments: different densities, a density gradient, or with dynamically varying densities? And he's curious what will be the response in these various cases.
Yeah, that's basically the PhD of two current students right now. We have been mapping baseline behavior first in what I call zero information environment. So much of the data that you actually saw was collected in IR because we don't even want to perturb any light-induced effects, for example. And once we now have the baseline data, what we are working on is essentially many of the sets of cues that I described. The vision is to be able to take CTD data, which is the location in which we collected the cell. We actually do this currently. We collect every single parameter that's physical based on density, light, temperature, and the time of the day. And we incorporate that in the wheel itself to really measure a trajectory that would exist in the context of that specific day or that specific location. Because in the ocean, you can actually see lots of fluctuations associated with this. So this is really the very beginning of some of this work itself. And of course, one of the ideas that we have around building these feedback cycles is that these cells have a baseline behavior in a zero information environment. The only thing that we cannot switch off is gravity unless we go to ISS. But one of the things that would happen is that my hunch is that then as we add every layer of this information, we will find feedback loops that account for that information to choose where these cells want to be. I mean, the big picture word, I often open my talks this way, but I forgot to mention is that we've been calling much of cell biology that happens on the planet as plankton, but plankton as a word is fundamentally wrong if you think about what it means as wanderer. It means as if it's completely passive and at the mercy of the environment. And as cell biologists, I am very passionate about proving that wrong, is the fact that cells have remarkable behaviors that they have evolved, and they do actually know where they want to be in the ocean.
Yeah.
The ocean is not just a homogeneous bath of cells. cells have incredible capacity in choosing what types of environments they would like to be, because if they don't survive the evolutionary race.
Yeah. I think we can take one more, Jen, right? So this is question from Pintu Patra, is giving an example about myxobacteria form similar vortices, and also the ants use trail in a very similar way. So he's wondering if cyanobacteria have a similar mechanism for these spirals.
Yeah, I think so. I want to qualify two very important things. It is not just a vortex. It is a spiral with flux going both clockwise and counterclockwise. So it's fundamentally very different from structures that we have seen in active matter before. And it's important because you can think about the shear that's associated, that the cells are seeing maximum shear. They love going against each other. Now, the adhesion and the reason dislocation theory is actually a good theory associated with this, in which you saw that there were no large gaps. The only gaps existed at the tips of the defects. So if this is the two cells and then here's another cell, you can see that triangle would act as a defect, and that triangle is closed because of adhesion, because of mucus. Mucus following can happen in scenarios when the cells are not perfectly in contact, where they are making trails. And we see that often enough in these sets of structures, that these sets of trails are useful in a low density kind of an environment. But this spiral is essentially constant density. There are no density fluctuations other than these breathing structures. So mucus plays a role in this theory on the context of adhesion, because without that, dislocation theory would not apply. And I think the other comment that I want to make is, this is a unique structure primarily because of entanglement, which arises from the spirals themselves.
Awesome. It's okay, Jen. If you haven't found it, I'll take it. Next question is from Ashish, asking if there are a smaller number of spirals going clockwise, would they compensate by swimming faster to stabilize the vortex? That's really fascinating. Is that a behavior adapted for some sort of survival?
You know, I mean, going back to why I do this, sometimes we have to really understand the ecology better to be able to answer why I do this. I don't know whether this is a physiological behavior, because we are working in 2D. It's not that we compress them massively in 2D. They do like to form two-dimensional structures. And there are many energetic arguments that have been given for two-dimensional mats. So I think the version that I have been thinking about in my own mind, because I care about asking this question for why stromatolites are so incredibly stable, is that there are lots of advantages in light environments when you are in clusters associated with who gets enough light. Having this flux in a material, I like to think an analogy to a spindle, because spindle, the microtubules are flowing in and out while the structure remains stable. In a similar situation, in a structure like this, the filaments are traversing all aspects of the structure while the structure remains stable. Whether that enables the survival, much of that has to depend of very physiological and environmentally motivated studies that we still have to do. Now, the other context of thinking about speed, it's a little bit complex, and we show that in the paper that we'll post. When we map and measure the speed, of course, certain sets of length has a correlation to speed, because of course, these are chains of bacteria. So there is an incredible association with the context of length. But the other aspect is that there is slip, because going back to recreational mathematics, take a spiral and start rotating it, winding itself in, you will soon realize that without slip, you will go to zero and it will be infinite elastic energy, which is not possible. You cannot wind a spring, because of course there is elastic energy in these filaments. You cannot wind a spring to zero. And so when you do that, and you can play with this with your watch spring, if you wind it and you let go, it doesn't just unwind, it actually expands. So there is a slip associated with this, which is radial, while majority of the velocity that you see with your eye is actually tangential. But without this radial slip, you would make a paradox that the center of the cell has to go to zero, which will be infinite amount of bending energy. So the energetics of this is that there is energy stored in the bending of the filament, there is energy associated with activity, and there is energy associated with dislocations. And these three energies are being essentially exchanged with each other to lead to this type of a dynamics. And when we do the numbers, they actually have roughly the equal, in quantity, they're roughly equal. And this is why this phenomena is even possible, because, for example, the active dislocations have been studied in how bacteria form its cell walls. And in that case, there are lots and lots of these defects that jam because the motor proteins cannot pump enough energy to break, or in the other case of a scenario, the motor proteins building these cell walls might be too powerful that the dislocation energy doesn't matter. Mm-hmm. So this is really the energetics associated with this. And of course, the velocity is a really tricky question because the velocity is not just in radial directions.
And Pradeep has a very connected question. Can the number of dislocations be related to the cell geometry?
Yeah, the number of dislocations is very simple. Because dislocations are conserved in our system because of active dislocations, dislocations don't disappear, but in the time frame of multiple days that we study the system, the filaments don't break. And actually, I forgot to mention, filaments also don't grow in this time scale. So the length is also constant. And so when you make this entangled structure, you can think about it that every tip of a given cell will have to have a dislocation. And since every filament has two tips, you will have two dislocations associated with it. So the number of dislocations is roughly twice of the number of filaments, ignoring the outside peripheral filaments. And that remains constant. And it is a fundamental parameter. So if you were to now build a structure like this with lots of little filaments, you will see the number of dislocations increase. While if you make a structure with very few, I forgot to mention, you were watching dynamics of filaments, roughly hundreds of filaments in there. And some of them were literally, 3,000 microns, while the filament dimension is only in the width is a couple microns, 3 to 5 microns sometimes. So these are incredibly high aspect ratio structures as well.
Fascinating. All right, I think we have the last question unless anybody else pops up with more. So Neva is asking about what factors stabilize or destabilize the spiral behavior.
What factors destabilize? So I think this is really the crux of what we discovered is spirals have this notion of a topological trap that will always stabilize a spiral in 2 dimensions. And that just comes from entanglement. And you can play this game that the early set of a structure can be unstable if you haven't put a clockwise and a counterclockwise spiral together. But because filaments come from outside and start joining, and there is a very beautiful, Jülicher did this long time ago in active filament theory, that filaments that are long enough have a certain probability that if they go down and interact themselves, that is the origin of a spiral. So if a filament is moving and all of you have played that game of nibbles. I'm assuming some of you have played the game of nibbles in which there is a little worm that you're moving around and it's very hard to avoid self-intersection. That's what leads to a framework for the first spiral to form. But then when a new filament comes, it is literally a topological lock. The new filament can come in 2 directions. It can either come and wrap around counterclockwise or it can wrap around clockwise. And if that geometrical chirality is different from when a new filament comes, you've already locked a structure. And then you can keep adding mass, and mass can leave as long as you don't flip chirality. But with probability, you see there's a 50% chance of getting either the same chirality or not. So very quickly, there are these locked zones. And what I love about this, and we are now starting to look in fossil records to see if we can find a hint of this in fossil records. But again, fossil records, the challenge is that it's very difficult to find filament level because those are ghost fossils. You don't actually get to see cellular context. There are some fossils in which that exists. But the idea is that there is a fundamental stabilization that comes purely from topology, which is not possible in any vortex, active vortices, this is fundamentally different because of entanglement due to spiral geometries. This phenomena is only true for spiral geometries because those geometries cannot actually interweave.
Okay, I think that is all of our questions. So with that, we'll start to wrap up. So again, thanks to everybody who stuck around for 20 minutes past the hour. Thanks to everybody who's still here. And for anyone watching on YouTube or watching later, we'll see you next time. And I guess we'll see everybody in September or October. So again, reach out if you're interested in presenting. And
Please do
thanks
that,
again
yeah.
for being part of this